All right. Let's get started because it normally takes a minute for me to say random stuff before I even get there. I will jump over some slides, which in the past students have loved, in that I'll say in 10 seconds what you need to know about this one since we're running behind. So just focus on my words there. So today, this is about carbs. This is the single time there's kind of times where I'm like, this is the most important thing I'm going to tell you. If I could give like one actionable thing that's the cheapest, this is that lecture. So I'll talk to you about, for a quarter of a day, what can you do to help a patient? Long story short, it's increased fiber. And that doesn't include, that doesn't beat out some things that are free, such as like sleeping or eating less or, that sort of stuff. Okay. And some photo bombs. Okay. And let me make sure I have the chat window up. All right. We're off. Whoop, not quite. So here are the objectives, and actually, since I'm a little early, maybe I can get the notes because I forgot to do that. So I forgot to do that for the last one, but here are the table of contents I like to do for the online notes. Okay. Objectives. So this is the general formula for a carbohydrate: carbon, some amount of hydrogen, H2O times some amount. And here is the breakdown of the different diets. So yellow is fat, red is carbohydrate, blue is protein. The typical Western diet: too much red, low glycemic index, modified Atkins diet, medium chain triglyceride diet, and classic ketogenic diet. So you see they all have a lot more fat and less carbs. So metabolism is complex. We'll get into glycogen and starch, these storage forms for carbohydrates. So we talked about storing of fats before, and now we're talking about our sugars. And in metabolism lectures you'll hear from me later on, we basically have three energy sources: carbohydrates, fats, and proteins. Proteins are this unique one where they're also on nanomachines, so they're different. Fats and carbs we basically use as energy, and so we have these storage forms. This whole purple box is an FYI. You don't need to know it, but here's listing the different monosaccharides: 3 carbon, 4, 5, 6, and 7 carbons, and where they are. The only thing you need to memorize from this is this red box of the 6 carbons: glucose, galactose, mannose, and fructose. You don't need to know the structures, but I'm going to be talking about those and the differences. So here are these different words to describe different chemical structures. It can take a long time to go through this. I'll try to do it in half the time, just so you can read the slide and understand it. These are all isomers. A stereomer is a type of isomer. Within stereomer, and a stereomer has the same sequence of bonded atoms. In this, I tried to show hands so you could understand what's going on. We're all used to enantiomers being like the two hands that can overlap, but that are mirror images, right? But can't overlap exactly. Well, enantiomer is a type of stereomer in that it's exactly the mirror image. A diastereomer is all the other stereomers. And stereomers have that same sequence of bonded atoms. So if you wrote C number, H number, it would have the same number, and they would be connected to the same. Isomers have the same number, but they're not connected the same. Then we're going more similar. Epimers, they have a difference at only 1 carbon. And then specific to carbohydrates, there's anomers, which the 1 carbon that has a difference is at the anomeric carbon, and those are termed alpha and beta. All of that was to get me to anomers, which we'll talk about. And so here are examples of epimers. You don't need to know any of this structure. You can use this or not, but you will have to know the anomeric carbon, which ones don't have them, and a disaccharide. I'll talk about it. So again, you don't have to know these structures, but at room temperature, these carbon chains can form cycles and open and close a cycle. So they're linear and they're cyclic, okay? And so when they're written like this, you can see that there's a business end in that there's this carbonyl that can be attacked. When they're closed, it doesn't look as much like there's a business end, but the anomeric carbon is kind of what attacked that to make the ring, okay? And the take home here is if there's an anomeric carbon available, it can be attacked. So it can have, for example, that sugar could attack or be attacked by a protein, and so you could have a protein that then gets a sugar added to it, which you don't want. We'll talk about that. That's called glycation. If there's not an anomeric carbon, it's not going to react. So we term these monosaccharides having no glycosidic bond. The glycosidic bond is between these two sugar molecules, okay? A disaccharide has one glycosidic bond. So here's a disaccharide, one sugar, two sugars, di. Oligosaccharide's a little looser. It's kind of more, 2 to 10 glycosidic bonds. And then polysaccharides are very big, 10 to hundreds. And so the reason disaccharides are important is we have specific enzymes that work on disaccharides to break them up. And we have different enzymes that work on polysaccharides too for digestion. All right, so this is one thing you guys unfortunately can't get like last year. And when I have you guys in class, I normally bring in these, I don't know if you can see me, these different fibers, and I have them for you to just add water for and use, and to see what's the difference, what are good, what are bad. I'll talk about it. Okay, so how do we store sugars? We store them as glycogen, this very branched chain, okay? And they have the linkages, the glycosidic linkages. I'll back up for one sec. Where do I say that? The term alpha or beta terms is a term of where is that oxygen coming off that's gonna form the glycosidic bond. If the oxygen, again, you don't really need to know the structure type stuff, but if the oxygen on the left here that I'm hovering over, the blue one, is on the same side as that methyl group coming off, it's beta. If the oxygen is on the other side from the methyl group coming off, it's alpha, okay? And the reason that's important is we have enzymes that can work on certain alpha, beta, and not. So if you go down to glycogen, we have alpha 1,4 linkages and alpha 1,6 linkages. The alpha 1,4 is kind of this long part of the chain, and then the alpha 1,6 is less frequent and it causes branching, okay? And we have enzymes that can break that apart. So as we get sugars, we build, as in a fed state, we build up the glycogen, and then when we need to break it down, we can chew them off, okay? Fiber in the form of cellulose has beta 1,4 linkages, and we cannot break that down, okay? So we can't get energy from cellulose. There's also this starch fiber, and that can be termed soluble and insoluble. And we have gut bacteria that can digest some of this. The insoluble generally passes straight through us. The soluble bulks up, soluble is amylopectin with 75, starch has 75% amylopectin with branches, and amylose 25% without branches. And I'll talk to you more about fiber in a little bit. Okay, do we have time for this? If you want, in the next 10 seconds, rank order, I'll come back, what cereals you eat, and I'm gonna show you, that still should be open. Here is the cereals listed and how much fiber they have. So they're named pretty well, right? The things with bran have a lot of fiber. I spent so much time in the cereal aisle always trying to find these healthy cereals. They're all pretty much not healthy, right? It's a lot of sugar. So if you look at how much fiber you're supposed to have, if you're a male less than 50 years old, 38 grams per day, and if you're a woman, 25 grams per day. Once you're older, it drops to 30 and 21 grams for men and women respectively. So usually each year I start to preach about, okay, having a certain amount of 5 or 10 grams of fiber in a cup of water each day. And then a student's like, well, why don't you just eat more vegetables? And clearly, Mediterranean diet, increasing fruits and vegetables is better. It's very hard to prescribe that to a patient and have them do it. But for basically the cost of a quarter a day, you can have someone drink their fiber, and it has massively good consequences, health benefits that I'll tell you. Another question I get is, well, I'm going to talk to you about how when in our body, when we store fiber, it swells with all this water. Why doesn't cellulose do that? Well, cellulose, it was that long linear chain. And so even though it has all these hydrophilic groups, which normally would be swelled with water, they actually pack right next to each other. And that's actually why it's kind of rigid and stiff. And it doesn't swell with water the way that our stored carbohydrates do. All right, so let's talk about what happens with diet, and then if you add fiber. So in these graphs, the dashed line is just your normal diet, and then the solid line is if they've added fiber. And basically all of these things going down is good. So your fasting glucose goes down. When you don't eat, your glucose leaves your blood, whereas diabetics have high glucose in the blood. Your fasting insulin goes down. If you're fasting, you shouldn't have insulin. That's good. Your glycated hemoglobin goes down. That's all good. And so here's a table I've kind of boxed for you the things that I want you to see. They're talking about insoluble and soluble fiber. The best fiber seems to be psyllium. That is Metamucil or various brands that are generic from Costco and such. And if you look at these effects, cholesterol lowering, improved glycemic control, helping with constipation, diarrhea, irritable bowel syndrome, it does all of those. And for basically 10 cents a day, you can have 5 grams of that and get this effect. Now, what's interesting is looking at the dosage. So this is grams of whole grains per day. So not just fiber, but the whole grains have fiber. And if you look at around 100 grams, so translate, maybe 10 grams of fiber or something, I'm not sure the translation, but if you look at stroke and cancer and all-cause mortality, stroke kind of does plateau at about 100 grams of whole grains per day, but cancer keeps going down. So this is relative risk. So relative risk of 1 means no change. A relative risk of 0.8 means you have a 20% chance, reduced chance of it. A relative risk of 1.25 would be a 25% increased chance of it. So for stroke, you've dropped by about 30% chance of stroke just by having 100 grams of whole grains a day. Cancer drops and keeps dropping. I wish I found papers that continue this further. Where does cancer stop? But you can reduce your risk of cancer over 20% just by having whole grains, or you can just supplement with fiber. All-cause mortality, this means dying from anything, that drops about 30% and keeps going. So it's very cheap and very easy to do, to have fiber. Oh, let me back up a second, see if you guys were healthier. Oh man, this is not worth it, but, and what were your responses? Cheerios, which is not bad. I will say I recently bought Honey Nut Cheerios, which is delicious. And then when I looked at the label, it's all sugar. Life, I like that Captain Crunch. Yeah, these are all unhealthy. And then below the yuck line was the brand, total brand. All right. So you guys are not outside the norm. That's just, how things are. All right. Is this correlation? That I assume you're talking about, which is a great question. I love it. This graph right here is not correlation. This was dosed, I believe. And then these are correlation. I almost always, have the studies. So you can look up those studies. I don't remember. I don't remember if this was metadata or not, but there have been, my guess would be that this is multiple studies from correlation. But there are causation studies to know you dose with fiber and it changes your gut biome. Long story short, I don't know about that for this graph. Okay. All right, go ahead. And I told you that you should have 5 grams of fiber. I do 10 to 20 grams of fiber a day, probably 10, 15. It's hard to drink that much, not including what I eat. So go ahead and click which you think is 5 grams of fiber. This is of the Kirkland natural psyllium fiber. Okay, 5 seconds. 4, 3, 2, 1. Very good. So, well, I wouldn't say very good. It's this one. It's kind of a trick question because if it was, I don't know if you can see what I'm holding up, but if it, depending on the fiber you use, and again, I show this normally in the class, if you're using one that's for the powder that has real sugar in it, then 5 grams of fiber would be somewhere in between these right two. It would be about this much. And so, for example, I'm holding the one that has sugar, and its serving size is 24 grams. And that's because it has 16 grams of added sugar. So most of what you're doing is drinking sugar. Whereas and that's a bad one. And then the one that's more just fiber, its serving size is 6 grams. So 6 grams versus 24 grams. And the soluble the fiber you get in them is the same. Now I will say that the one that has the 6 grams serving size has fake sugars in it, and they have their own issue. But that's most of what you can buy. Okay. All right, we'll skip over this. The take home here was how many ways can you combine sugar molecules versus amino acids? If you were making a polypeptide for the amino acids, there's basically one compound you could make. If they were radiocarbon labeled carbons, if you knew the difference between 2 alanines, you could make 2. But glucoses, you can make many because there's so many OHs that can attack the business end of the other molecule. Okay, so the take home there is that sugar tagging is numerous, and we're starting to have drugs that are proteins that are glycosylated, and we have to make them look exactly like the way they're made in our body. And there was a question about how well we did that. Do the generics do it as well? The update from last year was that the generics seemed to do it okay, so those drugs seem to work well. Some of the nomenclature is if you're talking about something that's a mix of like protein, something with carbohydrate attached, whatever's at the end of the name is more of it. So for proteins, a glycoprotein is mostly protein with some sugar added. The glyco is a sugar part. A proteoglycan is a mainly sugar molecule, multiple sugars connected with some protein on it. A glycolipid is mostly lipid with some sugar added to it. Okay, and the proteins can have sugars added on NH groups or OH groups. Those are called N-glycosides or O-glycosides. Let me see how we're doing with time. Okay, not bad. So early on, it's hard to talk to you guys about stuff because, it's a chicken and egg. You guys, we haven't taught you enough. So I'm going to give you the most basic example of metabolism, waking sleep cycle that I can. So you wake up and you are in a fasted state because you have been fasting all night. Okay, and so you have glucagon and you then eat, and when you eat you secrete insulin. Okay, and so if you have insulin, you are in a fed state, so you're increasing your glycogen. Glycogen is the storage form of your sugar, and that gets stored in the liver. If you're in the fasted state when you wake up with glucagon, you're increasing your glucose, you're tearing down glycogen to use it as sugar, okay? I don't know if I'll get this to another slide, but for every pound of glycogen that you synthesize, it binds 3 pounds of water. So when you fast for even 24 hours, you deplete your glycogen, and if you lose 2 pounds, let's make it easy, that's a lot, but 2 pounds of glycogen, you would lose 6 pounds of water, and you would lose 8 pounds. So that's why it's very easy to look like a diet works very well instantly. But then if you dieted for 3 days and then the fourth day you had some food, you'd rebuild that glycogen and suck up and reserve more water, maybe have dark urine for a little bit, and you'd instantly bounce back up in weight. So that's a helpful thing to know, to explain to people as they diet, that you've heard the adage that it's water weight earlier on. It is, and this is why. It's because the glycogen binds that water. So here's a graph showing what's happening. In the blue, we're looking at the standard diet of breakfast, lunch, and then dinner. The ideal diet for losing weight, all things being equal, would be to basically have a big breakfast and that's it. You'd fast for 24 hours. I can talk to you about that. It's very hard to do socially because we want our meal to be at night, but food sets your circadian clock for in the morning with the light. We'll talk about that more later. So anyway, at nighttime you were fasted, so gluconeogenesis is ramping up. That's you're making glucose. Then you have breakfast and it's down. You have this little peak between each and then it starts going up at night. If you look at glycogenolysis, that's down overnight and then it's up between each meal. Okay. So you have these endoglycosidases that hydrolyze poly and oligosaccharides, so big stuff, and then disaccharidases that break apart di and tri, so smaller sugar chains. And so here's your memorized slide to know maltose, lactose, and sucrose. What is maltose? Maltose is a disaccharide of glucose plus glucose, and there's an alpha linkage. Lactose is galactose and glucose disaccharide with a beta linkage. And sucrose, which is table sugar, is glucose plus fructose and has an alpha beta linkage. You don't need to know the structures. I'm just showing you them here. And then the glycosidases that work on them are obviously named maltase, lactase, and sucrase. Okay, and so this is why lactate can work for people that are lactose intolerant, because you are breaking up the disaccharide for them. So here's a table showing the enzymes. The one unique enzyme is isomaltase and sucrase, because that is made as a single enzyme and cleaved. So if you have a deficiency, which we'll talk about in hitting both of them, you've lost both of those enzymatic functions, right? So I'll leave this I'll read through one line and then you can read this. Isomaltase breaks the alpha 1,6 bond. You know, you don't really need to memorize these. You need to know what they do. The bond cleavage, for example, the compounds that it cleaves are isomaltose, and it produces maltose. Whereas sucrase cleaves sucrose and makes glucose and fructose. Should we know the linkages? You need to know the linkages for, can we metabolize them, and how do they, what is the effect of tests? I'm going to talk to you about a Benedictine test, for example. Okay, so what is happening when we eat stuff? If you eat a piece of bread, it tastes sweet. Can anyone put a C in the comment chat if you can tell me why bread tastes sweet? Okay, Alexis, go ahead.
Is it because we have amylase in our saliva that starts to break down carbs?
That's exactly correct. So alpha amylase right here starts breaking down the carbohydrates in bread into a simple sugar that we can taste. It's not like the bread has glucose on the outside that we're tasting. So you start to break down some of the carbohydrates. It goes down to your stomach. In the low pH, the amylase is inactivated. Then it goes down and you have alpha amylase in the pancreas, so you start digesting again. You also have these other enzymes. You're breaking everything down into individual sugars, so there's no disaccharide bond, and then you can absorb those sugars, okay? Now, if you are absorbing all that, what comes out? We're basically a tube from the front end to the back end. What's coming out the other end? Well, since we're short on time, I'm just going to go for those. Cellulose, right? The undigestible stuff. Here is that slide that I've shown you before. Now we're going to get, now we're talking a little bit more about absorbing of those sugars. So the GLUT1 is in the blood-brain barrier. GLUT3 is in the neurons. These are all sodium independent, okay, GLUT1 through 14, and they transfer glucose. GLUT2 is in the liver, kidney, and pancreas, and then GLUT5 is the outlier where it is working on fructose in the small intestine and testes, okay? And we've already talked about the SGLT1, that is the sodium co-transporter. Remember we talked about that for, is Gatorade the best thing to drink? And basically you want something with sugar and sodium in it to, if someone's dehydrated. Okay, very board testable. Just memorize this little GLUT table now and you'll save yourself time later. Okay, so here we have a disaccharide in our intestine. Oops. It goes in and it is a significant molar amount, right? So from the water lecture, we know that water will get pulled into the intestine to kind of change the concentration to try to dilute this disaccharide, right? So water's going into the intestine. Then you have, this is for things that you fail to digest, right? These are disaccharides that have a bond we can't break, some sort of soluble fiber, but the bacteria in our gut biome can digest it. So when it digests it, one, it will multiply, but also it will release out 2 and 3 carbon things, carbon dioxide, hydrogen. That is more molar things, right? You're breaking apart, just like we did that math before for sodium chloride versus magnesium chloride. You're ending up with a new number of higher things. So what's that going to do? That's going to pull more water in, okay? And so what is all of this doing? This is diarrhea, this is cramps, this is gas, okay? So this is what happens when you eat too much that's going through you that you can't digest, but your gut biome can. So now we're going to get through the, we have like 10, 15 minutes left, the genetic diseases, galactosemias, fructosemias, lactose intolerance, CSID, and diabetes. Some of them I'm going to go through very quick just because the slide's there and I just want you to know the genetic defect and what's going on. So here are the galactosemias. You have, follow this chain from the bottom left, galactitol to galactose 1-phosphate. The main one to know is classic galactosemia, this big red star. 1 in 30,000 people have this, newborns. It causes developmental delays. You can't have breast milk because of the lactose. If you back up one chain in the chain, there's a different disease, galactokinase deficiency. So this, so the first, each of these is a deficiency in the enzyme, right? So the classic galactosemia, the deficiency is in this galactose 1-phosphate uridyl transferase, GALT. Another disease, galactokinase deficiency, obviously the galactokinase is deficient. There, in the boards, or you'll see, a classic presentation would be cataracts, because if this is broken, you back up galactose. Galactose quickly becomes galactitol, and that aggregates in the eye. So a patient presents, baby with cataracts. This could be one of the things. On the right here, you guys can't come to my office, but I have this metabolic map printed out big on my office, and so I might show you sometimes where that is. I believe we have the metabolic map as a big file in foundations folders somewhere. If not, I can get it to you. The main reason I wanted to bring this up is that for the USMLE, they were planning on giving this on the boards. I think that idea has kind of failed last year because they had an issue with people that are colorblind not seeing the colors and such. But the take home, which is still important, is that the fact that they were going to give this to you shows the type of questions that are on the boards. No question is answerable by looking at the metabolic map and seeing this enzyme goes from A to B. That's just rote memorization that they would give you. The COMLEX might be a little different, but for the USMLE and COMLEX in general, they're higher level questions than that. So as you're studying and thinking about boards, know, okay, the question's not just going to be this rote memorization, it's going to be stuff like this. Galactitol backs up in galactokinase deficiency because the cataracts of the galactose backs up into galactitol. Okay, and then the last one, which is much rarer, is galactokinase epimerase deficiency, which is related to this enzyme UDP galactose epimerase. And so the diet is no lactose or galactose for this. HFI, hereditary fructose intolerance, is deficiency in this fructose 1P aldolase or aldolase B. It's 1 in 20,000 births. It causes severe hypoglycemia, vomiting, and fructose, sucrose, and sorbitol can cause liver failure and death. So the treatment is to remove those fructose and sucrose from the diet. And then the last one, essential fructosuria, is a lactofructokinase. It's very rare. Once you're at less than 1 in 100,000, I think of those as quite rare. Autosomal recessive, and it's a benign condition. But you can have fructose accumulation in the urine, which might come up in a second. So you guys have probably heard this story already about lactose intolerance, but in short, we have this, all humans are born with producing the enzyme lactase because we get nutrition from our mother's milk, breast milk, and so we can break apart lactose with the enzyme lactase. As you stop drinking milk, you stop making lactase. So if a baby continues to drink milk, they will continue to make lactase. In addition, we have populations that don't have lactose intolerance, and that evolved with domestication of basically of livestock, of cows and goats and stuff. And so here's a heat map of the genetics of people that can still consume lactose, and so it maps to where cattle were domesticated in Northern Europe and in Saudi Arabia. This is my sad baby emblem because it's like the saddest of the diseases in my mind. Congenital sucrase isomaltase deficiency, CSID. This, remember from that chain linkage, was that enzyme that's two enzymes made in one and then it's cleaved and so you have two functional enzymes. Well, people with this disease are lacking both enzymes. So the single polypeptide is normally cleaved, making sucrase and isomaltase. And so these people can't do either of those. So think of a baby not being able to drink a malted, or not being able to consume significant carbohydrates. The treatment is sucrade, which is a basically sucrose from yeast. And so you can do a bowel biopsy, which is the gold standard of diagnosis, and the results would come back with decreased enzyme activity and acidic stool and hydrogen in the breath. Can anyone tell me where hydrogen in the breath is coming from? Type C into the chat. Go ahead, Monica.
Would it be from their gut bacteria?
That's exactly right. Yep. Yep, very good. So you're getting stuff, you're not being able to absorb it, the gut bacteria can, and they're releasing hydrogen. Okay, so how do we test for some of this stuff? This is back to that thing about, is there a reducing sugar? So again, when you have a monosaccharide, there's no glycosidic bond, so all of those can open up, the ring can open. If you have a polysaccharide, they'll only react as a reducing sugar if there's a free anomeric carbon. On the next slide, I just give you a cheat sheet table of what's what, but there's this test called the Tollens test or the Benedict's reagent, and it's positive if there's a reducing sugar, okay? Now, what is the issue with reducing sugars? So for example, type 2 diabetics have sugar in their blood. Their sugar's too high because they're not responding to insulin. Insulin would normally be like, we're in a fed state, taking the sugar, but you're not responding, so they have high sugar. Because they have high sugar, some of those sugars have anomeric carbons and can react to proteins, and so your proteins that are going around that have nitrogens or oxygens can get glycated, and that's where the sugar gets attacked to them in a non-controlled way. Glycation is bad, so this is in red. Glycosylation is a controlled version of putting sugars on the protein. So we have proteins that as they're translated, sugars are added, and that can cause different recognition. So that's desired, and they're in certain places, and that's good. Glycation is good. Glycosylation is good, controlled putting of sugars on proteins. Glycation is bad. And this is what I've just told you. So the hemoglobin gets glycated. So hemoglobin is A1C, and you can measure the glycation of that hemoglobin. And when you measure it, you're measuring basically how much was glycated in the last 3 months. It's not like an instant sugar reading. It's have they had high sugar over the last 3 months. And so for diabetics, you'll have over 48 millimoles per mole. Of note, you'll see in the next slide, fructose is different in that fructose in the urine is a false positive for this. So you could have essential fructosuria, and that's because fructose has that business end. So here are the non-reducing sugars: sucrose, trehalose, sorbitol, mannitol, fructose, and then the reducing sugars: maltose, lactose, glucose, and all monosaccharides. And again, here's the note about fructose. Itself as a ketone is non-reducing, but in the alkaline environment, it slowly converts to glucose. So in fructosuria, a reducing sugar is found. And then you can have these glucose sticks with glucosidase that would test specifically and rule out fructosuria. So one of the last things is that for the rest of us that are not diabetic, we still have problems with glycation in this things called AGEs. I love this acronym. So AGEs are advanced glycation end products. And so as we age, we have proteins that get glycated and shouldn't, okay? Those things that get glycated are called AGEs. So there's not one age. And in fact, sometimes people refer to AGEs as, let's say I have protein A and it gets glycated. And when it gets glycated, its pathway changes and something, protein B, increases. Some people might even call B an age because it's an advanced glycation end product. But more usually and kind of more succinctly, the age is the thing that actually was glycated, okay? Now we have this receptor called RAGE, receptor of AGEs, and it binds to multiple AGEs. And so how would that happen? That's because RAGE can detect, it has a protein structure that can detect sugar compounds, so it can detect generically proteins that have sugars added. So there's many AGEs, many proteins that get glycated that bind RAGE. This is all bad because when RAGE gets bound, it is pro-inflammatory, so it increases inflammation, TNF alpha, IL-1, IGF-1, these things that you'll hear a lot about, which can cause atherosclerosis and increase vasoconstriction, so all these bad things. And that's it. Finished basically 1 minute ahead of time. I know I went fast, but I kind of succinctly told you what to focus on, and go ahead with put a Q if you have any questions that I'll record. Okay, Alexis, go ahead.
So where it says diabetes greater than or equal to 48 millimoles per mole,
Yep.
do you know about which A1C level that would correlate to, or do they correlate?
Oh, sorry, I'm muted. Sorry, I was muted because of all the dings. I have a slide, I forget where this is, I'll show you in some future lecture, where I show you the actual distribution of glycation of hemoglobin and the relative amount. So just wait for that.
Okay.
Jose, 6.5, what does that mean? Oh, are you answering a question? All right, no more questions. I'm signing off. Also, these should now be recorded directly into Panopto. So once I hit end, it should take 20 minutes or however long Panopto takes, and then that recording will just be live there. Yep. All right, I'll see you at the week recap next Tuesday, I think.